All right, today we are diving into something that sounds like it's straight out of science fiction. We're talking about a completely new way to engineer biology, to basically take the driver's seat of evolution, using something as simple and honestly as elegant as light. I mean, just imagine for a second. What if we could guide the evolution of life's most basic building blocks, these things called proteins, not over millennia, but with like the flick of a light switch? It's a huge question, and it's the core idea behind this absolutely groundbreaking new technique. But before we can really appreciate just how brilliant this new solution is, we gotta understand the problem scientists were smashing their heads against. It all starts with a really powerful method that had, well, it had one big nagging limitation. Let's call it the always on problem. So, for a long time, scientists have had this tool called directed evolution. It's basically a way to put natural selection on fast forward inside the lab. The best analogy, really, is to think about ancient farmers. They'd pick the best crops, the strongest ones, and only plant their seeds for the next season. Scientists were doing the exact same thing, but with proteins. But here was the catch. The old method was kind of a blunt instrument. It was all about applying constant pressure, always, always, always selecting for the protein that was the strongest or the most active. And the result, well, you got these proteins that were basically stuck in the on position, permanently. And that created this huge disconnect from how things actually work in nature. See, in our bodies, proteins are incredibly dynamic. They have to be. They're constantly switching on and off, they're responding to signals, they're juggling multiple jobs at once. But these lab-made proteins, they were static, one-trick ponies. And by forcing them to be always on, scientists were actually stripping away all the other cool functions that make proteins so useful in the first place. And this is where a team of researchers at EPFL had, well, a true flash of genius. They realized the goal wasn't just to select for strength anymore. They had to select for dynamics, for the simple ability to switch on and off. And they found their answer in light. They named it Optovolution. Pretty cool, right? Using light, or opto, to guide evolution. And here is the key difference. Instead of that constant brute force pressure, optoevolution applies a dynamic, time-based pressure. It guides proteins to evolve the ability to switch between different states, all while they're still inside a living, breathing cell. So you see, the whole mindset here has shifted. The goal is no longer about creating the strongest protein. It's about creating the smartest protein, one that knows how to turn on and off at exactly the right moment. The entire system just becomes a simple pass-fail test based on timing and control. Okay, so how in the world did they actually build this test, right? How do you use a simple beam of light to ask a cell, hey, is this protein doing its job correctly? Well, let's break down how they pulled it off. First things first, they needed a guinea pig, and they chose one of the most reliable workhorses in all of biology, budding yeast. Yep, the very same organism that's used to make beer and bread. It's so well understood, which makes it the perfect little factory for an experiment this clever. Okay, so get this, because this is where it gets really ingenious. First, they totally rewired the yeast cell. They linked the protein's activity to a molecule that the yeast needs to live during one part of its life cycle, but that same molecule is toxic in another part. Then, step 2, they flash pulses of light to tell the protein when to be on and when to be off. This creates the simple but totally brutal test. If the protein gets the timing perfect, switching on and off with the light, the yeast cell survives and divides. But if it messes up, stays on too long, or doesn't turn on at all, the cell either stalls out or just dies. The only ones left standing are the survivors, and they contain the new, superior, perfectly timed protein. And check this out, that whole life cycle for the yeast, it only takes about 90 minutes. That means every hour and a half, an entire pass-fail test, an entire round of evolution is completed. It's this rapid, automated protein factory, just running and running and running. So the theory is fantastic, right? But the real question is, does it actually work in practice? Well, the results are what really matter. And honestly, they're pretty astounding. 19. Just let that sink in. Using just a single starting protein, the team used Optovolution to generate 19 completely new versions. Each one was unique, with better properties. Some were more sensitive to light. Others were less leaky, meaning they stayed off better in the dark. But they did not stop there. They started achieving things that were considered really, really difficult before. For instance, they evolved proteins that could respond to green light, not just blue,
which opens the door to controlling different genes with different colors of light. They even made a red light system better, so it doesn't need this extra chemical to work anymore. And then they did something that is just mind-blowing. They evolved a protein that works like a tiny biological computer. It's what's called an AND gate. That means it will only turn on a gene if and only if it gets two different signals at the exact same time, a pulse of light and the presence of a specific chemical. This isn't just a switch anymore. This is decision-making, happening at a cellular level, evolved on demand.
So yeah, this is way more than just a cool trick you can do in a lab. What we're looking at here is a fundamentally new chapter in synthetic biology. It completely changes our ability to actually program life itself. You know, as the researchers themselves put it, these dynamic protein functions, they're at the very heart of how biology works, how it senses the world, how it makes decisions, how it stays in control. And now, for the very first time, we have a tool that can evolve these incredibly complex behaviors whenever we want. So what does this all mean for the future? Well, the possibilities are just massive. We're talking about building way smarter cellular circuits, developing tools that could be controlled by a whole rainbow of light colors, creating all sorts of new biotech applications. It even gives us this amazing new window to look back and understand how these complex protein behaviors might have evolved in nature in the first place.
And that leaves us with this really huge, tantalizing question. Now that we have the power to evolve biological computers inside of living cells, the issue isn't really if we can program life in more complex ways anymore. It's what will we choose to program life to do next?